over to Joe. So take it away.
All right. Thank you so much, Peter, Christine, for wonderful introductions. Thank you, Selmy and Michael, for all the work you did all these last few weeks and before I even joined that I didn't know about. And thank you especially to everyone for coming. One of my role models, I'll show you his picture in this presentation. He works at a company called NanoString and invented some cool technology. He starts off every presentation he makes by saying, "This is so fun to have everyone gathering here for this purpose." It reminds me in Plato's The Death of Socrates, they're saying, "Hey, why are you, the night before you're supposed to drink the hemlock, says, why are you doing all this philosophizing? You should be with your family, saying goodbye." And he said, what else could one do in the time before sunset? So we have, the virus, the election, and here we are thinking about how to help the future build better engineers and scientists. And I think that's wonderful. So thank you. So I'll share my screen. Let me know if it comes through properly. That look okay?
Looks great.
Okay. So I'm calling this mapping cell communities. We were writing one of our grants and trying to come up with a way to put in context why it's so important to look at maps of groups of cells rather than individual cells. And we hit on this idea of community. We since learned we weren't obviously the first people to think of it. But what you're looking at here is a somewhat stylized picture of a microfluidic chip. And I'm hinting at what's to come at the end of the presentation to try to keep you interested. And it looks a little like a neon light with this effect on it. And they call microfluidics chips in analogy with electronic chips. And that analogy bears good fruit if you think of, instead of electric current flowing, liquid flowing. So chips are tools to move liquid around in very precise ways. So my company, as Christine mentioned, is AtlasXomics. It's a very unwieldy name. I'm working on getting it changed. But we're extremely new, as Peter mentioned. We are based on a technology invented by Dr. Rong Fan, a professor at Yale, who has also been involved in BioPath. And he's a wonderful, incredibly smart and inventive person. And we owe a lot to him. So here we go. So mapping cell communities, here's kind of a broad overview of where I'll go with this. Why would you want to make maps of cells? What are these maps and what do they consist of? And how does DBIT, which is the name of the technology that Rong invented that we are commercializing, how does it make maps of cells? And that's when we get back finally to microfluidics chips. So I don't know if everyone here has played chess or learned chess or even taught chess, but if you think back to learning chess and realizing how important the interactions are between individual pieces, it might help you put in context why maps of cells are so important. So on the left, Black has made some not great moves, but at least he's still alive. Whereas on the right, Black player has made probably the worst first two moves he can possibly make and is now dead. And the difference between these two pictures, that one Black pawn, are you able to see my mouse?
Yes, we can.
Yes.
Okay, great. That'll help as a pointer. So this pawn here, being here or here, makes the difference in Black's life or death. And so positioning matters a lot in chess. You need to know more than what does the queen do. You need to know where is it in relation to other pieces in this chessboard. So when you're learning to play chess, you have a bag of pieces, somebody explains to you what they do, maybe you're reading a book, then you think about, okay, here's a particular piece, here are the moves this piece can make, and you do this for all the pieces. And finally, at the end, when you're actually playing, you're looking at a map of where the pieces are. And that's how you interact with the other player. So individual chess pieces, you can make an analogy with biological cells. There's different types of cells. They can make different moves, which really means they can make different proteins and perform different functions. And in 2019, Nature decided to call single-cell multimodal omics the method of the year. Let me just explain. Omics, kind of like genomics. We're talking about the profiling all of the DNA molecules in an organism. Transcriptomics would be all of the RNA molecules. So generally just an entire science is what they mean by omics. So multimodal omics means measuring multiple entire groups of things in one experiment. So if you look at this analogy and then look at this method of the year, you might ask, what will 2020's method of the year be? I don't know. I'm hoping it's spatial single-cell multimodal omics, but I'm extremely biased, so we'll see. So the tagline we kind of came up with is that cells live in and are influenced by their communities just like us. So you need to look at their community. If you don't even know how to profile their community, you can't begin to tell their story. So this guy, Joe Beecham, that I mentioned, is a super inventive guy. He invented the machine that does this experiment on a napkin in a hotel. So he's kind of like, a Tesla type person. And this, I'm going to explain on the next slide where a tissue section comes from, because I did not know before February. But this is a tissue section. This is a map made from a tissue section by depositing fluorescent probes onto the tissue section, having them bind to the molecules of interest, and then taking a picture with a microscope. So this is an optical method. And in green here, you're looking at cancer cells in a tumor. And in these other colors, purple and pink, you're looking at the immune system attempting to attack the tumor. We're looking at a battlefield map. And if you don't look at this map, if you just notice, in bulk that there's a bunch of immune cells and a bunch of tumor cells, you're missing a big part of the picture. At least that's the thesis. We've yet to cure cancer with this approach, but you can see if you were to look at this without the map, you really wouldn't know very well what's going on. So there's an amazing organization called the Institute for Systems Biology. There's some great people at it, including Leroy Hood is one of the fathers of gene sequencing back in the 80s and 90s. If you go to this website, they speak much more eloquently than I do about how all these different networks work together. But the key point here is that in order to make sense of these cellular networks, you have to look at the molecules that make them up. Because really the way you know which cell is which is by looking at the RNA and protein molecules they're working with. You can do a little with just how they look, but not all of it. And this fellow, James Heath, is actually one of the advisors of Dr. Ron Fahn, who invented our technology. So also an amazing person. He's been involved with Caltech and does a lot with protein detection and microfluidics. So if you go here, you can learn more. To this website, you can learn more about them. Another notable visionary in this field is Aviv Regev. She has been at Broad Institute. She's actually going to Roche as the head of research at Genentech, but she helped found this group called the Human Tumor Atlas Network. So cancer is not solved. This group decided to take a step back and say, we need better maps. They actually want to make 3D maps of all human tumors, profile the cells that make them up. So you can see there's momentum building and demand coming for ways to make maps of all kinds of cells, including cancers. Now, this also applies to other applications, Alzheimer's, ALS, virtually every disease, because humans live in 3D space. So every disease that attacks them in a spatial way is you need to make a map to help approach a cure. So because I'm going to be talking a lot about tissue samples, I just want to briefly go over what that is. So if you cut a piece of a person out and embed it in wax, cut it into very thin sections, kind of 5 to 10 microns thick. A micron is a millionth of a meter, and then mount it on a glass slide. Now you have something that you can put in a microscope, and people will typically do stains with it, especially pathologists. This is called a hematoxylin and eosin stain, which helps them distinguish the nuclei and membranes of cells. This is a very typical workflow for looking at what's going on in somebody's body. You don't only need to do this with human samples. You can also use mice or mouse embryos, zebrafish, insects, plants, anything. So this is how people look inside a 3D block of tissue. If you go to this video, you actually get a really good overview of this entire process. So I like to learn from YouTube, so try to include the links wherever possible. So once you have these tissue sections, you want to profile molecules on them. You can do what Joe Beecham and Nowstring do. You can stain them with colored molecules and look at them in a microscope. The more expensive the microscope, the better. Christine might know more about this than most of us. These microscopes can be very expensive. You have to train to use them for a very long time. And if you're looking at things optically, you're kind of limited in how you only have a certain number of colors, and they start to overlap when you try to do too many of them at once. So if you want to do 3 molecules and give them each one of the 3 colors, that's fine. If you wanted to do 30, they would all start to muddle together, and that would be difficult. So people realized you can do something called next generation sequencing. You can take a tissue section, and instead of delivering colored probes to it, you can deliver DNA probes to it. You can write down position information in DNA, attach it to the molecules you're interested in, and then sequence them in a next generation sequencer. It's a new generation of techniques. It was kind of kicked off for spatial RNA measurement in 2016 by a group called Spatial Transcriptomics, which was purchased by 10x Genomics. They now have a market or a total enterprise value or value of their business of around $15 billion. They are growing incredibly fast, and they're the public company to look at if you want to look at the companies in this field. There's also some other academic technologies. There's one out of Broad called SlideSeq. There's our technology called DBitSeq. They're all better at different things. None of them are the absolute best, but there's some jumping off points here from our researcher. Let me know if you have questions here. I will go a little more into detail about how the tagging process happens. I have some very detailed slides in the appendix, but it's super interesting how this works, and the notion of writing information into DNA is kind of the key idea here. So, but why now is next generation sequencing coming to the fore? You may have heard of the Human Genome Project. It took 10 years. It cost about 10 cents per base, the AGCT bases of DNA, to sequence one human's entire genome. And Gordon Moore was he was an engineer at Intel, one of the leaders of Intel. I think in 1965, he posited Moore's law. He said the number of transistors on a CPU would double. I forget the period of years, but since then the industry has pretty much followed this law or this prediction. Next generation sequencing since the early 2000s has accelerated, has become cheaper and faster, in a way better than Moore's law. So you can see now, starting about 2016, it all of a sudden became economical to sequence an entire genome in one day, doing it for about 10 cents per tens of thousands of bases. So we're talking about thousands of dollars instead of tens of millions of dollars to sequence a person's genome. This is getting to the point where you could, for an individual patient, sequence their genome for 8 cents. So you can see why genomic techniques are taking off now. So in DBIT, this is probably a crush of information. And I would love to go way more in detail, but we would probably run out of time, and I would probably fail to explain it properly. But basically, we deliver liquids onto a tissue section in a very controlled manner. So first, we flow liquids in a microfluidics chip, and I'll show you how that works in a few slides. We flow liquids in one direction, kind of like the rows of a chessboard, and those liquid could have a colored probe. They could also have a DNA probe that labels the row number. First row, second row, third row, etc. You can attach that probe to a molecule in your tissue section. You can then take that chip off, put another chip on that flows probes vertically, and stick them onto one another, kind of like Lego blocks. So after this second flow, in each of these squares, you would have a row number and a column number attached to a molecule of interest, like an RNA molecule. So once you do these two flows, you grind this tissue up and put it in a next generation sequencing machine, you can actually reconstruct a map of the molecules that you attach your probes to. So you didn't take this image in a microscope. You attached row and column numbers, wrote them out in a table, and then reconstructed a map like that. So it's a super interesting alternative way of taking a picture of a tissue section. And on the left here, compared to these RNA, this is a pan-mRNA, means all the RNA molecules. The redder the square, the more molecules are detected there. These are all proteins. So DBIT is kind of unique, and it can do the entire transcriptome and some proteins on the same tissue section at the same time. This is a bright field image, meaning passed light through the sample and looked at how it deflected the light. This is a hematoxylin and eosin stain, showing you the cell nuclei. So you overlay this digital reconstructed image with the optical image you take with a microscope, and you try to see if you can discover something with it. So Rong did this in his data. He noticed that the developing eye in an E10, this means a 10-day-old mouse embryo, here's the developing eye cup. People have seen this optically for a long time. They know when and where it is. This was the first time somebody could see it in its RNA profile. So this is kind of like somebody saw a star with a telescope, and then somebody else pointed a radio telescope at the same star and said, hey, that star is also really bright in this other imaging modality. So this was, does this lead to cure cancer? No, but it says, hey, we have another way to look at things. And maybe we can see things sooner here. Maybe a tumor developing would show up sooner in its RNA image than it would in its optical image. That's what we're hoping for. It's yet to be proved, but that's the goal.
So, Joel, that is actually a tissue, and how thick can the tissue be?
So the tissue sample itself could be centimeters in size. After they section it on a cryotome, this section here can be between 3, 10 microns thick. So a human hair is around 80 microns thick. So it's extremely thin. So you're really creating a 2-dimensional slice out of a 3-dimensional object. And it's so thin that if you think about human cells being around 10 microns in size, and it varies around there, but you're hopefully looking at a single cell thick layer of tissue. So in this particular image, these squares are 10 microns by 10 microns. So you're looking at a cube of tissue that's 10 by 10 by 10 microns. So it's around the size of a cell. And if you look at this intersection profile in fluorescence, you know, these are not perfect squares, and that's because you're looking at the boundaries of individual cells. So Rong likes to call this a cellular resolution method. There's a big push to get to single cell resolution. You would have to get a little bit better seeing than this, but it is, we call it cellular. It's kind of a compromise. One of the weaknesses of DBIT is that it doesn't have subcellular resolution. These techniques that rely on optical frequencies, their spatial resolution is limited by the diffraction limit, which is more like 1 micron, depending on the microscope you're using. So it's a little weaker in terms of spatial resolution, but it's much stronger in terms of how many molecules you can look at the same time. So this is a combination map of all RNA molecules. If in say a mouse embryo we detect 8,000 genes or 20,000 genes in total, you can look at a map of each gene individually of the entire genome. So it creates an enormous data set. And how to analyze this data set is, you know, with machine learning and a profession unto itself. So when I was teaching myself biology in February, it helped to think about analogies, and one that I use to remind myself of how cells operate is a library analogy. So if you imagine that you need to write a paper and you're going into a library which organizes information as words and books on shelves, you say, okay, I'm interested in cancer. You photocopy, or say viruses, you photocopy pages from books on viruses, not the entire book, but certain sections of it. You write your paper using those pages as source material, and then your paper goes out into the world and cures coronavirus. And please do this, or encourage your students to do this. That would be great. This is a comic from Lee Hood's and Jim Heath's organization of somebody slicing a virus in two. So maybe a nano soldier would be a good method. I don't know. And I love Calvin and Hobbes, and I searched Calvin and Hobbes typewriter comic, and this one came up. One of my favorites. So in a cell, which doesn't store information in books on shelves, it stores it as information inside DNA. And this is really useful to think of DNA as just a way of writing down information. And this is how Rong invented this technique, this protocol of writing information as DNA probes. You know, it's just a way of storing information. It's how our cells do it. During transcription, where DNA molecules RNA molecules will copy certain portions of the DNA. That's kind of like photocopying pages from a book. Translation, ribosomes, this big green guy, move along the RNA. They kind of trundle along and synthesize building blocks of life from the information on the RNA. Then the proteins go out into the world. They don't cure coronavirus, or maybe they would kill a few coronaviruses and so on. So that's helpful to me. I'm sure that everyone here is much more experienced than I am at teaching this concept. So if you have any comments on it, please feel free. So I showed you this comic. I showed that we're sticking barcodes to get to each other like Legos, but how are we achieving this flow of reagents? And the answer is with microfluidic chips. And I like to think of them as highways of tunnels, you know, group bundles of tunnels. So this bottom surface is flat glass or tissue. And above it, we have a tunnel made of this material, polydimethylsiloxane, that will guide the flow of fluid on top of the surface we're laying the chip on. So this is a 3-inch by 1-inch rectangular block of polydimethylsiloxane. It's a great material because it's squishy, it's elastomeric. You could drive a truck over it, and I think it would still work, though I haven't actually tried that. We did not invent it. This is invented, I'll show you the group's name, Whitesides in 1998 at Harvard. So you load liquid in at one end, say into these inlet wells here. This chip has 50 inlet wells. each of these wells is connected by a path, a circuit element, all the way across the chip to its outlet on the other side. There are 50 inlets. There are 25 here and 25 here. They come together. We would place the tissue section here. We call this the region of interest. So we have 50 rows. So these rows here, there's 50 of them. And then these columns here, there's 50 of these. So we would take the first chip off, place the second chip on, load 50 different barcodes, flow them through, and now we have intersecting rows and columns. Since there's 50 rows and 50 columns, we're actually building, when you stick them together like Legos, we're actually building 2,500 unique barcodes on top of the tissue. And that's the power of Wrong's technique, is that you're multiplying your input reagents. And this is an incredibly economical way to make many different reagents. The reagents, if you make them separately, cost about $10 each. So this would be $25,000 worth of reagents. But if you can do it with 100 reagents, now it costs a lot less, more like $1,000. So this is really a way to kind of democratize doing this kind of mapping. If you look at other techniques such as Visium and SlideSeq, they don't have squares made of intersections. They have individual spots that they print onto glass slides. So each of these spots has to have its own unique barcode. Visium has 6,000 barcodes. And Visium has a lot of advantages, but they've had to really have some very advanced manufacturing. I'm sure Christine would know how to do that specifically, to get 6,000 unique spots just by printing them directly. And in fact, if you look in how they do it, they actually use microfluidic chips to print those slides. So it's a really, really cool technology. This is a single layer microfluidics chip. There are ways to make much, much more complicated chips that I have no experience doing. And so this is you notice none of these channels touch each other. They're all separate units. So this is kind of this very simplest chip you can design. But even then, it's still an amazing tool. So here's a little diagram or the schematic. This tissue section, which is 5 to 10 microns thick, this is a mouse embryo, looks like around 13 days old. And you can see here the channels coming in and then moving left to right. And there's 50 of them. So you have kind of solid PDMS touching glass here, then a tunnel, then more solid glass as a wall between channels, then a tunnel, and so on. These tunnels, this distance is 25 microns, and so is the height. So there's have square cross sections. And then they lay down first on blank glass, then on the tissues. So they have to kind of mold themselves to this mountainous profile of going from glass to tissue. And that's kind of the biggest challenge of this, is how do you make that transition without leaking liquid out the sides. So we use a clamp to press the PDMS chip down on top of the tissue, which is on top of a glass slide, which is on top of a clamp. This clamp happens to sit in one of our microscopes. And then you pull using a vacuum. You place a vacuum over all of these holes and pull them through the chip. So in this case, this is how we made a picture of alternating red and green. So if you were to flow this through, this was food dye actually, then you would get alternating red and green in all of these channels. I think I have a picture of that later on. And then these springs are here to kind of equalize the pressure in these four corners. This clamp actually was a miserable failure. So we've gone through about 7 clamp designs so far. So it's kind of interesting how much this profile of pressure pushing down on this elastomeric block can affect the results. So here's another image. These were 50 micron channels. So they make 50 micron edge squares here. Actually, this is not, this is one of them. So because we overlay the results we get on top of the tissue section and take a picture of it, we end up with a bunch of pixels, which is amalgamation of the words picture element. And I was getting frustrated calling these pixels and also calling, the elements of this picture pixels. So I came up with the word tixel, which is a tissue element. And at first everyone groaned and complained, but now, I'm hoping to get everyone to change the name of the company to Tixel, because AtlasXomics is kind of a mouthful, but we'll see. So Tixel, you heard it here first. How do you make these chips? So I mentioned this name, George Whitesides, his group at Harvard in 1998, and they developed it all in the 20 years since then. They call it soft lithography. So lithography, generally, making small patterns. They call it soft because the material they use, PDMS, ends up being elastomeric. It's an amazing technique, and I'll show you. You can do it in a kitchen. You do not need a nano or microfabrication facility for most of it. So here are the steps. You take your 2-part resin, you mix it together, you take the bubbles out, you put it in an oven, you cut it with a knife, and then you punch some holes in it. And have behind me the machine that we use to punch the holes, or the tool. So just to show you, I think it's easy to think of biotech, oh, everyone's in a white coat using a sequencing machine or a microscope and so on. But, more than half of our company does what I'm about to show you. And we're only 5 people, so there's not very many people. But you take your 2-part resin, you mix it with a high-tech mixer. This was an espresso frother I bought on Amazon. That ended up being the best tool to actually mix it. The only difficult part of solid lithography is obtaining this thing right here. This is a wafer of silicon with a polymer deposited on it using photolithography. So that you cannot do in your kitchen. You need a CO2 laser and a collimator and hundreds of thousands of dollars of equipment. So we pay Yale $200 to make this for us. So once you have this, you can make many, many molds from it, dozens. Then you need to replace it. But once you have this, the rest can be done in the kitchen. So you pour your resin on top. As it flows and settles, it'll adopt the shape. So this is kind of an inverse image of the chip. It flows over those features. So here on that wafer, this is a raised feature. It's actually square. But so when you mold your polymer on top of this, that raised feature will become an inverse of that and become the tunnel. So you can see here, these are actually, a fine texture on top of the silicon. Since you've mixed it, kind of like making whipped cream, this is actually, this one I've removed the bubbles for before I poured it, but when you use beaters to mix cream, you incorporate a lot of air, so you have a lot of bubbles. You put it inside of a vacuum jar to get the bubbles out. They call it degassing. That takes like 45 minutes at a kind of 20 millibar vacuum. You can see it progressing. And then if any of these bubbles survive and end up at the interface between the wafer and the PDMS, you'll get a defect in your chip and avoid where there shouldn't be. So you really want to get all the bubbles out. Dust can also do that. If there's dust at this interface, if it's floating in the middle of the PDMS, it doesn't matter very much. But if it's on the wafer and the PDMS molds over it, that will also create a defective chip. So dust is expensive to get rid of. And Christine, again, knows much more about dust removal than us, but we kind of are hoping right now that we're not unlucky with where the dust lands, because it's too expensive for us to have a clean room. So if you had a clean room, that would be a little bit better. But kitchens work. The point about this technology is if you have this wafer, you can do it in the kitchen. And I would say probably 95% of our chips are not ruined by dust. They can be ruined by other means, though. So you bake it in an oven between 60 and 100 degrees Celsius. This was a mold we made out of acrylic. You can also just use a Petri dish. Actually works better than a fancy acrylic mold. Then after it's done baking, that takes between 2 hours and a day. You kind of cut out the part you want. So you can see on this wafer, I actually have the horizontal chip, we call it the A chip, and the vertical chip, we call it the B chip. So you've got to cut out the square and then also cut between them. And probably the single most important ingredient to our company's success is Scotch Magic Tape. It protects the chips from dust. It lifts dust off the chips if they're not molded into the resin. If you place it on top of the micro features, it highlights them for you, kind of with this relief pattern. Without the tape on, it's very hard to tell which end of the chip is which. This top is blank, and the bottom has the micro features on it. So without the tape, it would be very hard to tell. You can see here how starkly it shows you the features. Now you're not done yet, because see this is supposed to be a hole, but it's not. It's only a 25 micron deep well right now. We need it to be a hole to go all the way through the chip, so that when we load liquid from the top of the chip, this is the blank side of the chip here, the microfeature side is against the tissue. We need to be able to pipette liquid in there. So we have to punch a hole through the chip, and the hole has to match up with these outlines, and it can't intersect any of the other channels, because then we would have barcodes mixing where they shouldn't. So this is actually pretty hard to do by hand. So I ripped a stage, an XY stage out of a microscope, and connected it to this machine, which is behind me here in my apartment. And so I can finally control the spatial position of this punch with relation to the holes. If you want to see the holes very well, we're talking about kind of less than 250 micron clearance. So there's a lot of eye fatigue involved here. So if you shine a bunch of light on it, it gets very glary. And if you take polarized sunglasses, you can get rid of some of that glare and also make use of the fact that reflected light is polarized. And if you're reflecting light off of the wall of the tunnel, it's actually polarized in a different plane than light reflected off the flat top surface. So you can actually get a pretty good highlight of the channels that way. So I thought that was neat. I played around with filter paper, but it ended up that sunglasses, which were designed to do this while driving, are pretty good for this. And also, you know, they make you look pretty awesome. So we need help with that because we're all stuck in masks in the lab. So that's fun. So the kind of the tools we use on the bench after we do this molding and punching, we use a microscope to make sure we put the holes in the right place. We didn't intersect any of the highways, the channel highways. We use a much more expensive microscope, which can do image stitching. This is a motorized XY stage. You run a software program with the tissue inside of here to generate an image of the entire slide at once, which is difficult to do at high magnification. So that you can see the outlines of those stitched images here. And you can also do this after you do the experiment, after you've flown fluorescent barcodes. Here was a red and green barcode. So you can see that there's tissue here and blank glass here. And you saw that our barcodes stuck to the tissue in an alternating pattern, but not to where they shouldn't have. And you can see here too that the difference between red and green is not as stark. So in this area of this experiment, the red and green actually mixed together a little bit. So we have lots of failed experiments, some that worked. We're getting better, but it's a really interesting process. So just to loop back to our overview, we want to make maps of cells so we can discover new biology. You know, what are maps? They're spatial profiles of interesting molecules. And we make maps of cells with microfluidic chips, and you can make microfluidic chips with good kitchen practice. So that's the whole presentation. Thank you so much for listening. I just want to thank a few people. Wrong Fan, who has been involved with BioPath for years now, is an amazing person. He and his lab have helped us take this technology from an academic technology into something that would actually be a viable commercial product, which they're not the same thing. And there's an enormous amount of debugging and refining that goes in. They've been enormously helpful. So the paper for this is not published yet. It will be published in Cell in December. I'll definitely circulate that publication once it is published. Thank you so much to everyone here. Christine, Peter, Michael, Somi, you guys are wonderful. This is our CEO, Ken Wong. He's a really awesome guy. There's only 5 of us right now, so it's Ken and I, a few people, and a bunch of outsourced contractors. So we're really looking to grow quickly. It's difficult in this environment, as you know, but we're hoping that changes. And if you have any questions, please email me here. And again, thank you so much to you for listening, and hopefully you have lots of questions.